Good morning, one and all. In the session 7 of this complete series, we'll be learning about the biological pathway databases, that is Reactome. We'll be also going through the need of experimental methods to validate the predicted results. Understand that the protein is the major workforce in a cell, but proteins work together as a functional network. Okay, they do not work on their own. Every protein is interacting with several other protein partners to guide the function. Okay, so for example, if you're taking food, lactose, water, anything, different enzymes are expressed in the body to metabolize the molecules. Okay, and these metabolites, once the complete network guides the function, the overall implications takes place. But whenever I'm able to predict the protein network for species A, for species B also I can establish the similar network. Remember, I'm using the word similar, not same. Reason is I'll be using the homologous sequence information of species A onto the similar uh, protein partners of species B. Okay, so that the functional network of species A can now be extrapolated to species B also. Okay, but when I'm able to do it experimentally, what will be the strategy I'll be using? I can use the mutagenesis, site active mutagenesis, or like I can chop down the required enzyme to see the functional implications. Okay, I'm sorry. But besides all of it, the initially people have been using serial analysis of gene expression to analyze multiple different enzymes together, because all these mutagenesis techniques are only allowing us to check one enzyme at a time. But when I'm able to do multiple enzymes at one shot, that is much better. Okay, so Sage helps me in doing that. Likewise, the microarray strategy helps me to analyze several different enzymes together. Alright, so let us proceed further. So see, the Reactome pathway is majorly a free source and curated database. Curated, I mean, different source of information is constantly collected from various resources, including the research articles or the data mining through various pages of the NCBI, like DDBJ, all these servers. So it keep on doing the data mining from various sources to keep on mapping the probable homologs across different species, which are probably connected in different interacting networks. Okay, so through this, if we are able to uncover several different biological processes, including metabolism, signal transduction, cell cycle regulation, and even the disease control. Disease control, I mean, we can find the mostly connected protein within a complete network. Which protein is much more important, and then we can find the disease drugs against that. All right. Now see, if I extend the discussion to say, for an example, yeast and the human gene networks. For yeast, nearly 5,800 genes are there. So for this, if I'm considering one gene, one protein, it will be coming up 5,800 proteins. And if I say nearly 2 to 10 protein interactions are there for every protein, then it can be roughly be scaling up to 12,000 to 60,000 interactions, which implies of which nearly 10 to 20,000 are known. Means I'm able to somehow know from the experimental methods one third of the interacting protein networks, how the interaction is there, which protein is interacting with what other protein, and all the details. However, for the humans, nearly 30,000 genes, 40,000 genes are there, which now, as per the recent studies, can turn up to 40,000 proteins. And if the same statistics goes here as well, nearly 2 to 10 protein interactions are there for every single protein, which means 80,000 to 400,000 interactions are existing for human proteins. So among which only I am able to know 5,000, Now you can see it's nearly approximately 1% of the complete interacting network, substantially less. Now if I say, extending our old discussion, that every protein is not individual, many different proteins work together as a whole in a systemic network. Okay, so this systematic network of proteins can only be governed by the complete set of interacting proteins and not an individual protein. So until I'm able to map all these 40,000 proteins here, see, all these 40,000 interactions, I cannot get the exact network of how the interacting proteins work in a human cell. Okay, see, the picture is much more complicated. Now see, just as seen here, shown here, so many interactions are possible. But what is the ultimate reason of why so many interactions are actually possible? See the part. Biological literature, we have nearly 10 raised to 14 million documents available in PubMed, Google Scholar, different parts. But many of them are credible. But here I'm not saying credible or not credible. I'm just saying they are nearly there. DNA sequence data tends to 10 nucleotides. Gene expression data transfer 8 measurements have been there. But, see the but I have written here clearly. This but, I'm not at all emphasizing that they are credible or not credible again. But they are there through different sources. Here, gene polymorphism, tensor 7 known. Gene activation knockout studies, tensor 5. Okay, protein structure data, tensor 4 on the order. Order tends to 4 structures. Protein expression interaction data tends to 4 interactions. Protein expression data again tends to 4 measurements. Protein location data tends to 4 measurements. Now if I am considering the entire picture as a scene, from these available source of information I am able to map only 1% interactome. So it's really so much complicated. The picture is not so easy. Okay, so that is the extent of this entire interactome or entire discussion. Whatever we are chasing here, one protein is able to interact with several other proteins. Under what conditions we don't know. The interaction is really ternary, like only happening for a fraction of seconds, then nothing. Okay, and then it interacts with some other proteins. So under what conditions, what picture, the interaction occurs between the two proteins, and then suddenly it is no more there. All the proteins get dislodged. Okay, and then they find the new interaction partners to guide some other function. So it's all dependent on the protein functional network. Okay, now the to extend the discussion, so many consortium or the collaborations have been working here. See, the standard development and international associations, the gene annotation, okay, nucleotide sequences, micro and the gene expression data, the Human Proteomic Standards Initiative, PSI, the Bioinformatics, it is protein for the ligand interactions, the pathways, the bioreactome, and the biopex, the metabolomic standards. Okay, the complete systems model, that is, it considers the complete cell as one system. Okay, not proteins, not genes, but a complete cell as an altogether, which is able to guide certain interaction against certain conditions. And the conditions which are existing for one protein, the moment conditions change, the protein interaction may change again. And all this information is again covered by protein structure, protein sequences. And the Genomic Standards Consortium GSC is able to interlink so many molecules together. Now you can see the complete picture as such, through which all these are working together as a single team, but still we are able to map only 1%. Understand? So the picture is really big. Now see here, if I'm extending the discussion to just show you how the Reactome database looks like. Okay, see here. There is a pathway browser. You can check for different pathways right when you click here, or the pathway analysis. How different pathways are connected. You can analyze several different scores here. Okay, or how the pathways are like encoded in different species. You can compare the species or the expression data of different genes. You can check them all together. Okay, so see here, once you are able to see this reactome database, you can moreover also see the documentation of several different things. Moreover, like different tools which are considered for all this analysis, and the downloads are available here. Or if you have some questions, you can also contact the developers right here. See? And it helps us to analyze metabolism, signal transduction, cell cycle regulation, or even the disease. Okay, so several different utilities have been constantly there. Okay, and this entire reactome pathway, reactome database analysis use several different tools. See, in various formats it allow you to check, download the reactome data. See, the biopacks, SVM is systems by the markup language, is the programming language, just like HTML, SVML is there. Okay, the textbooks or the other formats. All these reacting in reactome data, you can download the data through in several different formats, and you can feed it for the other tools as well. Okay, so it's a beautiful service, beautiful server for various different analysis. All right. Now see the same part in different pathways, how different expression data is there, like how many genes are getting expressed, and what is the expression level of different proteins coming up in the pathway. So for that, see here, the same part, if I'm putting it here, see, extrinsic pathway for apoptosis, double strand break repair, like base excision repair, nucleotide excision repair. Okay, how many are there? See, 12 are present, different numbers coming up, matching proteins in data. See, and this is the part. See, how many of them are expressing actually expression level, and see this is the statistical part, lowest to highest. See, click on each of them, you can even get a heat map, but the different details are coming up. See, so the moment you click here, you get the expression data also. Now, see, to understand our discussion extended to like before moving forward, let us first check like how much we have understood so far. Okay, so answer the small questions. What does the reactome? What does the reactome database primarily focus on? Is it genetic variations and mutations, protein structure prediction, biological pathways and processes, and drug discovery and development? I hope you might have got the right answer. Yes, it is the biological pathways and processes. Okay, now tell the right answer. Which of the following best describes the purpose of the reactome database? Is it storing and analyzing genomic sequencing data, providing a platform for protein-protein interaction prediction? Remember, it is not predicting the protein-protein interaction. It is just able to analyze and retrieve the information from various databases together. I'm sorry. It is not at all predicting it. It is just a visual part just to extend whatever the data available in different part, it is just putting them together. Okay. One is curating and organizing knowledge about biological pathways and processes. Yes, this is the right answer. Offering tools for structural modeling of proteins. No, this is not at all offering the tools for modeling of the protein sequences. Okay. Now see the part. Visualizing biological pathways and their components is a key feature of the Reactome database. Yes or no? Yes, of course. It is just allowing us to see different data coming up in various things, various other databases. So only we are using the word curated database. Okay. Is the database, is the result reliable, or does it need to be experimentally validated? Okay, see, understand that I have been always been telling you that the data coming up here is just the extrapolation of data coming up in various species. Okay, so until you get an experimental data along with it, the results may not be credible, and you can always proofread it by using the mutagenesis experiments. Alright, now see the second session of this entire session is connected to the serial analysis of gene expression. This method is altogether connected with a specific requirement, that is, many times in a cell, different transcripts are always produced, different proteins are always produced, but I want to see is my required transcript produced or not? Only that's the question. So what will I do? I'll take a conserved section of this important protein and then check whether if the complementary sequences are present or not. I can easily match them. Like, suppose a small boy is playing in a sand and he want to see iron fillings are there or not. Now, rather than checking the iron fillings of the sand altogether, what he do? He take up some small magnet and then keep on rubbing up in the sand. What happens? All the iron fillings who are having the complementarity with the magnet, some interaction with the magnet, will always be coming and sticking it. Okay, so you can always get it. So by using exactly the similar rule, like scientists work, they use a complementary probe part as a sticking part. That this is there. Is something similar to it there or not there? That is only the simple question, which is worked out here in the strategy serial analysis of gene expression. So see, SAGE is a very powerful tool for the gene expression profiling. This is the keyword. Okay, for the biological research. It is a valuable tool for understanding the molecular base of biological processes, so that we can easily get in whether some proteins are produced under these conditions or not, and identifying novel genes and drug targets. How novel genes? Like multiple genes may have got the same conserved transcript. Okay, so if the motif is functionally present in several different proteins, even if I don't know some of them, I can exactly tap them. That under this condition, not only some protein X is produced, Y is also produced under the same case. So Y is somehow interlinked with the function of X, and I can map their functional network. Okay. So what are the applications? to study the expression of genes in different tissues or cell types, to study the expression of genes in response to different stimuli, such as drugs or hormones. Some proteins will be specifically produced to identify novel genes. See, just explained to you. To track the gene expression changes in disease. Okay, just like the microarray people do. We'll be learning that in this session. Okay, and phage is a powerful tool for understanding the molecular basis of biological processes and for identifying the novel genes and drug targets. Okay, so these are the few important examples and applications of how to use the sage, like and why it is so much important. Now see here, we have been knowing the one, like central dogmas, like the gene expression one gene one protein. See, the DNA to mRNA to protein. mRNA can also form the cDNA. This concept we have been learning from long time. Okay. Now understand that all these parts, like using by 10 to 14 base pair cDNA tag. Why cDNA? As we have discussed, like cDNA is already expressed. So now if I want to see what are the important transcripts coming up under these conditions, which are expressed, I'm sorry, I'm using the reverse transcriptase. So mRNA is converted to cDNA, and now that gene transcript is present, the cDNA is present. Now biotin-native primers are used to tag the cDNA. So the cDNA is already tagged with the okay. The cDNA is tagged with the biotin-native primers. Okay. Now if I am using the streptotin beads here, these streptotin beads are present in a column. I'm using a column which is having the streptotin beads, and I'm passing the solution through it. Okay. Passing the solution. So what happen? All these complete tags will be again bound here. Because biotin interacts with streptotin. Okay. Once they're interacting means I'm now through this complete streptotin beads. If I'm able to use streptotin beads, I can easily quantify how many tags are bound. Okay. So tags are then linked together, assembled together to form the continuous tags that have multiple pieces of transcript information. They are connected together. Okay. Then these long field tags are then cloned to the target cells, and on the basis of number of times a particular tag is observed, expression level of the corresponding transcript can be quantized. Okay, we can like approximately quantify how many times this particular transcript or specific transcript is expressed in the cell under these cases. So it is possible to reliably identify the proteins or find the relative concentration in the total mRNA. Okay, we can exactly know each and every part. Okay, just by a simple technique. We are using the cDNA tags, we are using the biotin primers, we are using the streptotin beads, and then we are just interconnecting all these short transcripts to form a big long part. Then once if I'm able to clone it together, I can exactly quantify what all transcripts are produced under these cases. Okay, isn't it interesting? So Sage allows us to check many different transcripts together. This is how the complete part goes in. See, you isolate the mRNA, okay, then add a biotin labeled DT primer. See, you add the primer here. This is biotin labeled. Then you synthesize the double-stranded cDNA. This means double-stranded cDNA. Okay, you synthesize it. Tag is attached here, like connected with biotin. The biotin is already present here. Everything is there. Now when I'm using the separated in beads in the column, see, these are separated in beads here. Okay. All this biotin is connected here. See, which this transcript is also connected. So now I'm using the cleaning with the anchoring enzyme. Okay, so that it generates a sticky ended part here. Okay, so the sticky ends are not generated, through which I'm able to intercalate all the individual transcripts. All the small transcripts will now be connected together in a single log big transcript. Okay. So now see here, I'm using the linker part here. I'm adding the linker sequences. I'm like getting this part, see, and with it tagging enzyme, see, blunt end restriction endonuclease, see. So all of these are connected here, see. Everything is connected here. All the small pieces are connected here. So see, now I'm using the T4 DNA ligase. See here. So now I'm calling each of the part as ditag, see. Linker, and this is the ditag because two tags are connected here. See? Okay. So it is termed as ditag. Now I need to extend all of this further. See? Linker, ditag, linker are then constructs are amplified by PCR. See? Everything, so many tags are now coming up. See? It became many tags. Now again I'm using the anchoring enzyme, see? To remove the linkers. Because now I need to now connect all the individual tags together before cloning. So there I don't need the linkers. Otherwise it will increase the length of the transcript, which I don't want. The entire game of linker was just to establish initially, so that I can get the transcripts initially at the large part to form the right text. But now I don't need linker. Okay. So now I again use the anchoring enzyme to cut down, chop out all the linkers. So see, the tags are now combined into longer molecules called concatenamers. This is an important part, see, the concatenamers. Now all the ditags are combined together to form a single log big part which is termed as concatenamers. See, all of them are concatenated into single log segment. Now small question, conceptual question for you. If I'm having four tags T1, T2, T3, T4, what is the order of them in the concatenamer? Can I find out? Think of it. You need to think and tell me how many possible concatenamers can be generated by using the four tags. Okay. All of you find out how many possible number of concatenamers can be generated by using the four tags. Or does it has got any order for it? Like T1, T2, T3, T4, or T4, T3, T1, T2, or T1, T2, T4, T3, anything. Or can I even find it out? So understand that at first place, for the first, when I'm putting the first part unit, how many possible like T1, T2, T3, T4, anything can come in. So for the first unit I have four options. The moment I go to second unit, I have got the conditional probability. One is already used. So left over is 3. So I can only put 3 tags. So 4 into I can only put the 4 into 3. Okay. The moment I go to the third place, I can then put in 2. The moment I go to the fourth part, I'm only left over with one tag. So 4 into 3 into 2 into 1. Okay, like this gives me like 24. So there are 24 possible concatenamers which can be generated by using the 4 tags. Okay, so all these concatenators are then analyzed in the computer. Okay, so understand that we are forming the tags, then we are linking the tag together, we are forming the die tags. Die tags are again combined together to form the big concatenators. They called together interconnected by T4 DNA ligase or the anchoring enzymes. Okay, but before cloning we remove all these anchoring enzymes so that we put in all together. Okay, and then we are only using the computer to analyze. So see here in this part, in this slide, I'm showing you the possible parts. See, here I'm showing the example of a concatenator. All these tags are connected here: tag 1, tag 2, tag 3, again tag 1, then tag 4. Okay, they can combine in any order. Now, in my previous example, only one option was there, but in the cell, multiple options are possible for each tag. Anything can come in. Okay, so then I need to quantify. See, the computer program generates a list of tags. Okay, and then tell me how many times each of the tag is produced in the cell. Okay, it is exactly quantifying. Okay, so this indirectly gives me how much times, like how many times the expression level was there for certain transcript. We know that all these transcripts are coming up okay, done. But now when we are able to find out that these transcripts are coming up, it has got a substantial impact on the overall survival and the functional like implications of certain key proteins. So what are these key proteins involved into? So for that, what we do is we check on the functional implication of each of these, the key signal, the key tag. Okay, so these tags we find out the function. Okay, these functions can be checked out through different databases. Okay, the different databases which are specially meant for the the sage part. Okay, specific databases are built for which each of the transcript, the functional implication is encoded there, stored there. Okay, we check out. See, transcription elongation factor 1 gamma, T complex protein 1Z. Okay, subunit, no match. We could not find a match for this. See, RPA1 RNA segment, ubiquitin protein, SPSF1 protein, NADH dehydrogenase, RPL21 ribosomal protein, L18A ribosomal protein S31, ribosomal protein S5, homolog M1, and see BGDNA GM12270. Okay, so there are certain these functions are present for each of these tags. Now understand that if this tag here, see, is expressed 99 times, unlike the tag which is present here, for which you could not find the match, or this T complex is only present 2 times. Okay, so it's got a diverse meaning. It has got a diverse meaning. What is this up to? So that is the entire implication of how to analyze the sage data. Okay, under what conditions, what things, certain transcripts are produced, and their expression level tells us a lot. Okay. Now, after knowing how to analyze the SAGE data, what is the complete protocol? Now, let us understand what are the merits and some limitations of the SAGE, of using the SAGE methodology. The biggest advantage here is understand that this overall gene expression patterns come up with the digital analysis. I can get the digital record here, see, completely. What transcript is expressed, how many number of times, so that I can do a comparative difference in the computer. It allows for the rapid sequencing analysis of multiple transcripts. How exactly? Because I'm able to integrate multiple die tags into single long concatenator. And by cloning the concatenator, I can easily identify how many transcripts are produced in a cell under certain conditions, and what is the expression level. So it allows us to rapidly analyze all the sequencing data at one shot. And because stage does not require a pre-existing clone, it can be used to quantify and identify new genes as well as known genes. Some genes which are quite similar to my old gene can also be taken up here. Okay, so there's a high chance that I can get some new genes also. Okay, depending on the dye tag. These are the major advantages. Now let us understand the limitations of using the stage method. The major limitation of using the stage method is its high cost. That is the we must sequence nearly 50,000 tags per sample. $5,000 per sample. See, so many tags are there. So for getting so many tags, we need to use a large quantity of proteins, large quantity of tags, like large expression level, large system, and after doing all of that, it goes for nearly $5,000. See per sample. mRNA levels and the protein expression or level always do not correlate. This is another major biological trouble. Many times mRNA is produced, but the protein is not seen. Okay, or the protein is only produced for like very less amount of time. mRNA is formed and got degraded. I could not catch it even before the reverse transcriptase. It could not be caught. It could not be caught. Okay, so these are the major limitations. Now, I have just shown you how to map the different tags to the genes, the functions. And for that, this is the website, see, the stage map. This is the stage map website, see. I can read its paper here, the complete biorepository, see. Okay, gene expression omnibus, see. All connected together. All the expression data coming up also takes up the input from here, or the stage also takes up the inputs coming up from the geo part. Okay, so they're always keep on getting connected, see. So see, this is the another tool. This is the another tool, see, the sage net. The complete expression data of various things, various genes can be analyzed together using the sage net. And these are further parts, see, on the PubMed also, see here on the PubMed data also, and see, I can retrieve my sage data. I can put the sage, like the tag sequence here, see, like Homo sapiens, and I can get the function, see. Total tags under what conditions certain tag is expressed is found, what are the involved interlinked papers to it, research articles, and what is the key function for each tag. Different analysis can be done also by the NCBI web server, and it's all connected together, see. So it's so beautiful. Now see, understand that if I extend this part further, like a big repositories have been built for analyzing the sage data. See, for human colorectal tissues, several different libraries have been formed here. See, NC1, NC2, CSCO2, SCT116, SW837RQ, TU102, TU98, ADA, ADB, this is adenoma A, adenoma B. See, the pancreatic cells. Likewise, this page was very long. I just shown you a snapshot here. Okay, I'm sorry. A snapshot is shown here, see, but this data is very big. Okay. Now, before we proceed further, let us answer these small MCQ questions to affirm our understanding so far. Which of the following is not an advantage of stage over other gene expression profiling techniques? Stage is more sensitive and can detect lower levels of gene expression. It is indeed wrong because even if some gene expression has happened and the amount of transcript is very low, or it has happened for just like a small turn amount of time. It cannot be caught even. Okay, because only when the cDNA reverse transcriptase is able to produce cDNA, only then SAGE can work. But if the mRNA expression level is already very less, cDNA will not be able to produce. We cannot produce cDNA. So the SAGE will not be able to give good results. Okay. The SAGE is more quantitative, SAGE is more versatile. Yes, both are correct. SAGE is less expensive. Yes, it's indeed correct. Serial analysis of gene expression stage is a technique used to determine the DNA sequence wrong, identify the differentially expressed genes in a sample. This is indeed wrong. We are not analyzing the differentially expressed genes or multiple gene expression at the same time. We are checking the expression of a single gene. Okay, whatever the required gene is there. That's it. Okay, measure the protein levels of a specific gene. Yes, this is the right answer. Analyze the structural organization of a chromosome. So option C becomes the right answer. Alright. So now understand the small conceptual questions. SAGE is a high throughput technique used to quantify gene expression levels. It's a high throughput strategy not to identify gene expression levels. We are only interested in identifying the quantity of a single gene under varied conditions. It may be there. Or detect some copies of genes which are quite similar to my gene or my transcript. Okay, the probe which is there. So this becomes a false. Can SAGE give wrong results? Yes. Okay, if we are unable to get the gene expression levels rightly, we cannot caught. Or if the my probe required 10 to 14 base pair cDNA tag is able to interact multiple different like cDNA segments together, it will give varied results for large amount of sequences, large number of DNA sequences, which may or may not have connection to my initial transcript, which I want to check for. Okay, so it can give wrong result, which I need to verify it later. Now let us move on to the microarray strategies for analyzing the gene expression levels. Now see how does the microarray strategy takes place? It has got all code like 7 different steps. First is the target design and selection. Like I want to do the microanalysis for a specific gene. So that specific gene, the target gene, I need to imprint it on the microarray chip. Okay. So for the microarray chip, I need to put the complete set of target genes already imprinted there. So that is the target design and selection. So before we do it, I need to first find out what should be the target gene under this condition, and one should once we find out that completely, so before imprinting it, I need to affirm that this is the right gene under this condition. Say suppose I want to check the breast cancer part. Okay, so some gene is which is specifically expressed only in the case of breast cancer, I need to take it, so that whose expression level tells me exactly about the breast cancer. Okay, or say some brain cancer. Okay, the brain cancer, some specific protein is produced. If I am able to detect that brain cancer specific protein, I can exactly find out that yes, brain cancer has occurred, and in these different cells it has got varied expression levels, so that I can extract some meaningful information. So once I am able to fix that this is going to my target gene, I can then print it on the microchip. The, then the RNA extraction. Now to once the chip is ready, I need to get the sample. Okay, I need to get the sample from the different cases. Say I want to check the same brain cancer part in the patient cell and the normal human. Patient and the normal human. So I need to extract RNAs from both of it. Okay. Once the RNA is there, I need to label it. Okay. Then hybridize and wash it so that the unbound RNA, unbound samples can be removed, unbound sequences can be removed. Okay. So hybridization once, because once I allow it, like the initial the my required sample DNA is bound onto the target design, the design gene sequence. It is already bound there because my target is already present on the microchip. Once I take the RNA extract and again convert it to cDNA and then allow it to bind here. Okay, there will be binding here exactly. Okay, but if some unbound part is there, that can be removed because hybridization means again binding of the like the labeled gene of target gene with the sample gene, then the scanning, and then the analysis. Okay, so now understand that all the scanning takes place is done. But once the scanning is there, it is the picture is taken up and then is analyzed through the system. Now see, let us discuss the key differences between the stage and microarray. Detect unknown transcripts. Stage yes, because I am taking a small transcript, cDNA transcript, which is able to interact with different other parts. As microarray, I am more interested in whether it is there or not there. If it is there, how much relative level of expression is there. Okay, that's the only part. So microarray cannot get the new part because it's a big part. Okay. Now see the quantification. It is the absolute measure here. Like how many transcripts are produced? 47, 49 are detected because it's all tags. Remember the die tags, and I'm able to form concatenated. The long many transcripts will be able to give me a more bigger concatenated. But here it is relative expression. So it's relative measure. Sensitivity. Okay. See how much smallest amount of unit it can detect. The sage is very sensitive. See, it is able to detect a very small amount of also one, two transcript as I've shown you. I've just shown you in the table. Okay, example table earlier that the sage data gave me only a single transcript or two transcripts, three transcripts. So I was able to pinpoint each and every transcript clearly. But in microarray, I cannot get it exactly like that. Specificity, moderate, okay, because the stage tag is able to interact with several other related genes as well. But in microarray, it's highly clear. It will give me exactly what I want, nothing other than that. Direct cost 5 to 10 times higher than arrays. See, 5 to 10 times lower than sage. Sage is really very costly. See? Now see the DNA microarrays. See, here two parts exactly the same thing. Now we are extending on to the probe and target. See, one which is already printed on the microarray is termed as probe, and the sample genes which are taking up from either patient or the normal or different cases from different organs are now termed as targets. Now see the probe is integrated on the slide by like by chemical reaction on the robot is called a microarray fabrication. So once we are fabricating the different targets or the key genes onto the part which are probes against which I want to do the complete check. Okay, because my microarray is especially designed to check the expression level of only this gene, which is termed as probe. Okay, I'm I'm doing the fabrication of this key segment probe. Okay, so the CDN of interest is bound onto the side so that the extracted DNA samples can be checked. See, it is a labeled cDNA cDNA is allowed to bind to genes of interest. It is a cDNA sequence because mRNA is not very stable. That's quite obvious. Okay. So now the probe is bound. The target part is set. Everything is done. So this is termed as microarray fabrication. Now coming on to the targets part. Now this is the sample data. cDNA sequence of all control and test samples are allowed to bind onto the probe. These sequences are usually of certain genes that are supposed to be controlling the cellular cellular response against certain stress conditions. Now see, these targets usually vary within the length. They are normally 25 to 60 bases, although it may be of 100 to 1000 bases long also. Now understand, if it is smaller target, it can bind onto the desired part, but if it is bigger, it may give me wrong results also. Okay. Now here I am using two dyes, Psi3 or Psi5. Psi3 is green. Okay. It gives me the green fluorescence. Psi5 gives me the red fluorescence. So the advantage is, if suppose in two conditions I am doing the microarray, suppose the normal cell and the mutated cell, or normal cell or the disease cell. Normal I can give Psi3. The disease part I can give Psi5 red color. So that once all the colors are present together in a single slide, single microarray chip, I can do all the cells together on a single chip. I can do all the experiments on a single chip. That's the entire beautiful beauty of this part. Okay. Now see, understand that once the complete probes are already bound on the microarray chip, here see, once I'm allowing the samples to come in there, the samples will be bound onto the probes. Okay. So the targets will be able to interact with that. So once somewhere the interaction is successful, this is termed as hybridization. The the cDNA hybridization is taken place, microarray hybridization is taken place. So once the fabrication is correct initially and the hybridization is done successfully, all the leftover parts will be like wiped out. Then you can wipe out all the leftover things and then you can scan the results. Okay, under the fluorescence. So Psi3 and Psi5 dyes, the green and red dyes will be giving me the signals. Okay, so the color will vary from Psi3, Psi5 and the mixture. Okay, the hybrid in the hybridization part, understand that this is a stable part where it completely forms the bond. This is unstable one because where certain part of the probe or the target is interacting but not completely. So only picking the right probe is very essential. Okay, as I have shown you at the initial part, screening the right gene and designing the right target. It's very crucial first step to design microarray. So see here, DNA microarrays attaches DNAs onto the slides and then capture the information about the set of DNA points. Okay, it will simultaneously measure the level of expression for a large number of genes. That is what we have been discussing so far. So see this case, normal part, tumor part, like I am doing the RT-PCR reverse transcriptase, I am able to get the part. Here also I am using the dyes. See Psi3 and Psi5, the green and the red part. See, and then once I am able to combine the part together, I am putting it all together. See, this is the microchip which I am getting lastly. After microarray fabrication with the required probe, I'm allowing the targets or the sample data to sample cells to bind, sample genes to bind, and then wherever it's bound, it's okay. Otherwise, I'm washing it out. Okay, then I'm getting different parts, see red, green, or yellow. See, depending on the relative concentration of these mixtures of these colors, I will be getting diverse colors in between the green and the red colors. Okay, which the system will be able to caught, will be able to catch. Okay, see, Then I am able to analyze this data in the as a heat map in the computer. See? So the relative abundance for spotted DNA sequence in the two DNA or RNA samples can be accessed by monitoring the differential hybridization of these two samples on the slide. Allows the expression of up to 20,000 genes simultaneously. See, that's the individual beauty. Even if 20,000 probes are present at some plate or some microarray plate, I can analyze all the 20,000 genes, how the expression levels have been there under these different conditions. Okay, so that's the entire part. So now let us discuss why this target design and selection is very important. See, this is the most important part here. Because this is ultimately going to decide what will be the accuracy of microarray. Okay, so we are synthesizing the oligonucleotides from 25 to 70 bases in length. Okay, two sequences complementary to mRNA of interest. This is very specific. I am fixing the right part, which is going to be very crucial in discriminating the right part. That this is normal cell, this is tumor cell, and this is the key protein involved as a discriminating element between the two. Okay, we need to avoid long A plus T or G plus T rich sequences, because otherwise they will be like binding here and there. Okay, to minimize internal secondary structures that may form hairpin or other loops because of this complementary repeating element. See, this is repeated residues, so we don't allow A, T, 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 C, C, G like that, because they will be forming hairpin loops. So to fix it properly, what I do is I design and select the right oligonucleotide oligos. Okay, these oligos should not be that are less than 75% identical to existing genes elsewhere in the genome. Whatever the part I am choosing, whatever the oligos I am choosing, whatever the key target I am choosing, okay, that probe should not be present elsewhere. Okay, greater than 75% identity should not be there anywhere. So I'm doing a blast against all the genes in the database for my required probe. So whatever the results come up, I check out all the results should be less than 75% identity for my initial probe sequence. Then I will be able to fix the right probe. Yes, this is the required probe I want. This does not have any complementary or any similar sequence present in the database with greater than 75% identity. For this, blast search is very important. Okay, because sequences with greater than 75% identity will be cross hybridizing here and there, and this will lead to confounding results. The results will be very disturbing. We cannot analyze. Okay, there will be mixed results. It should be erroneous. Okay. Now, before we go further, let us quickly see how does this Psi3 and Psi5 dyes are used, how the infected samples are properly analyzed in the microarray, and the similar parts. Okay, enjoy this small video.
Researchers first remove samples from both the uninfected plant and the plant infected with the virus. mRNA is extracted from each of the plant samples. The mRNA is then reverse transcribed into more stable complementary DNA, called cDNA. The infected sample is fluorescently labeled red, while the uninfected sample is labeled green. At this point, in many microarray experiments, the two cDNA samples are combined. The combined sample of fluorescently labeled cDNA is applied to the microarray chip. The cDNA from the sample binds to its complementary sequence of DNA bases on the chip.
We know that the pieces that are fluorescently labeled will stick tightly to the corresponding gene features, but they won't stick tightly anywhere else. So we rinse the chip off after we do the hybridization step, and then we put the chip in a laser scanner to electronically capture the data.
The chip is scanned with a laser, activating the fluorescent dyes in the sampled cDNA. A computer captures this information, calculates the ratio of red and green on each spot, which indicates which genes are expressed in the uninfected and infected plant samples.
Alright, so I think you might have enjoyed this short video to understand what we have been discussing so far. Okay, now see the seed in the microarray has been used in different parts, different conditions, different tissues or the same organism, like brain versus liver. I can do the check. Same tissue, same organism, like in treatment versus control or tumor versus non-tumor. Okay, different conditions. Same tissue, different organisms. Means wild type versus mutant. I'm mutating certain tissue. Okay. So in different organisms, how the things have been worked out. For the same tissue, I can easily capture. Or the time course experiments means at 1 hour, if I'm giving the drug, 1 hour of the treatment, 2 hours, 4 hours, 6 hours, 8 hours, 10 hours, like that. So in the time course experiments, I can progressively see how the gene expression data is changed for the same set of tissues. Okay, so it allows me to like completely evaluate the effect of treatment or the development under different stages. Okay, so what are the applications of microarray? There are numerous applications. Measuring transcriptome abundance as the initial part, cDNA arrays, measuring mRNA decay rates. Okay, because cDNA expression level will go down. So it is somehow interconnected with the mRNA decay. Okay, we can properly analyze. Identifying protein binding sites. Okay, under different conditions, we can evaluate the protein binding sites as well. Determining subcellular localization of gene products. Okay, that after some point of time, the some specific proteins are coming up only in these cases. Some specific organelles. I can easily see that only because of the expression of this gene, certain protein has gone there. Okay, so somehow some connection is there. So for all this analysis, this microarray analysis is very important. So see, when I'm scanning it, the part, the fluorescence using the laser detector, see, when I'm putting the laser on the chip, all these colors are reflected, the fluorescence, and I'm able to get this signal. So see, the Psi5 gives me absorption at 650 nanometer, okay, and the emission is at 670. Here 550 absorption excitation, and the 570 is emission. Now before moving further, let us solve the small MCQ questions to affirm our understanding so far. What is the primary purpose of microarray in molecular biology? Analyze DNA or RNA samples, detect genetic mutations. I cannot detect genetic mutations using simple microarrays. Okay, I need to analyze lot many of them to affirm. Analyze gene expression of protein interactions. Yes, the gene expression data can be analyzed. Okay, but under certain conditions, some genetic expression, some gene expression is going to change. Okay, and that can be caught. That some gene expression has gone down under some case, or something has gone up. The last one is determine the sequence of DNA or RNA. This is of course wrong. Which of the following best describes the purpose of microarray analysis? The microarrays are used to generate gene mutations. They do not generate the gene mutations. Okay. Mutations provide a method for DNA amplification. It is the for this the PCR is there. Okay. For amplifying the DNA sequences or to produce multiple copies of DNA. Okay. Microarray does not do all this. Microarray allows for simultaneous synthesis of thousands of genes or proteins. Okay, the C becomes the right answer. Okay, so the C is the right answer. Microarrays determine the sequence of DNA or RNA samples. Okay, that's the so the option C is the right answer here. All right. Now, before we go further, answer this small conceptual question as well. The main principle, the main principle behind microarray technology is hybridization between target molecules and specific probes. Yes, because of hybridization only I am able to detect. Because I'm putting the probe on the chip earlier, microchip earlier. Then I'm allowing the different targets to bind on it. Okay, from the samples. Can it give wrong results? Yes, when the somehow the aberration do not take place, or my probe initially was very small or long, it was having the large number of C's, G's, multiple C together, multiple G together, because of which the improper binding will take place. Hybridization will not be all throughout complete hybridization, it will be just small stretches of hybridization all throughout, the scattered hybridization all throughout. Okay, so this will lead to like internal hairpin structures, internal looping. Okay, so the small binding, improper binding can give me like the false results. Okay, so this is like a major, this can give me the wrong results. Before we go further now, let us analyze a few more things within the microarray strategies. Because microarray part is very complex. It involves a lot of strategies, lot of steps, wherein there's a multiple set of complete concepts. And in this complete session, I'll be showing you several different parts. Okay, so I hope you might have understood so far. So now let us proceed further. Understand that the microarray production part is here. See, the target design, selection, and printing. This is the slide making part. Because once I'm able to confirm that I'll be checking the expression level for certain this gene. Okay, then I'll be fixing the most important conserved part for that gene, and then I'll be printing it onto the microarray. So there's the slide preparation part. The experimental part involves the RNA extraction, which we'll be using as samples. The one that I've printed on the slide is termed as probe. Okay, so I am able to extract the RNA extractions, the labeling, and then I label it using Psi3 or Psi5. Psi3 for normal cell, Psi5 red for the different conditions, a mutant or the infected cell, like that. Okay, then I allow them, once everything is done, samples is ready, probe is ready, I allow them to hybridize. I put the samples onto the probe, they'll hybridize. So somewhere the proper binding is there, hybridization will be complete, indicating the complete binding, complete hybridization of the sample onto the probe. But okay, so somewhere improper binding is there, it may be there, but yes, we can escape all of it. So we'll wash out the complete leftover like sample part, sample sequences, and then I allow it to scan. Then I put the laser beam on it so that I can extract like what is the excitation pattern of these samples, these sample genes. Okay, the hybridization part. Okay, so I'll be able to extract exactly get like in within this sample what is the expression level for this specific probe. Okay, and then I analyze the data. Okay, within the microarray reader. Okay, like that the complete thing is there. As I have been discussing earlier, after putting the laser beam onto the sample part, microarray chip, it will emit a large amount of colors. Okay, so the green for Psi3, Psi5 red, and very different colors in the middle. Okay, so this I'll be able to detect here. See, so all these parts will be coming up, and then this will be correlated with the expression pattern. So this is termed as heat map. Okay, this is termed as heat map. All these colors, see, here certain readings are plus, certain readings are minus. Understand that these negative readings are not negative as such. On a whole, they are negative, once I'm taking an average. So compared to a big average, the expression level for certain gene under some condition has gone statistically negative, statistically lower than the average. Okay. Understand that this complete correction of the microarray values, complete correction of all the strategies includes a few important steps. These are pre-processing steps. What do they all mean is that before we actually analyze the values or to actually get the meaningful biological information from the values, we pre-process all the results, the macro results. This means image processing. Okay. Then to extract the red and green fluorescent intensities for each spot on the array. Then we normalize. Normalize means x minus mu upon sigma. Value minus mean of all the expression values upon standard deviation of all the expression values. So whenever the gene expression value for certain part is substantially higher compared to the average of all the expression values, the values will be more and more plus. However, if the gene expression value is substantially lower than the overall average of all the gene expressions, the value will be much more negative, as you have seen in the earlier table as well. The values were substantially deviant between the minus some value and plus some value. The negative reading and the positive reading, okay? But all this helps us to identify the outlier values. If we are finding that most of the values are confined within certain limits, but few values are substantially deviant, means these few values may be the outlier values. Okay, so we eliminate them. We exclude them for further analysis. We don't consider them. Then we correct for the experimental error and the systematic differences between samples on the same slide. Because if some samples has undergone the experimental error, there may be values. The the values typically change. Okay, which is undoubtedly like in what order, what things, like undecisive manner. Some values are drastically different, which cannot be analyzed. Why this has gone this way? Okay, so we again analyze. So the analysis is not only between the slides, is also between the values of within the values of a single slide also. Once we are confirmed that everything is done rightly, the normalization is done properly. Okay, then we go for estimation, testing, clustering, discrimination. After everything is done properly, and we have like eliminated all the errors, everything is done okay rightly. Then we come to the final part, that is the biological verification and interpretation. What do they all mean? All right. So see here, once I'm putting it all together, see, pre-normalization was something like this, see. But once I'm able to normalize it further this way, I'm finding that the average which was there is now this way. I'm able to get much more results properly. Much more genes are coming up, see. Here some mathematical error was there, see. Some gene has got expressed properly, but still I'm unable to get the result. Okay. Likewise, the number of values can go down also. See, here I'm getting many values, like many abrupt genetic expressions has been shown for several genes together. But here it is not like that. The moment I'm fixing the right average value, right things, so I'm able to normalize the values properly, and I can get the required results in a much more better way. So between the pre and post normalization values, like the number of values may increase, may decrease. Okay, so the person has to properly choose what is the required part and how to manage it. See, another major part which is causing the trouble here is the segmentation part. Means many times when I'm putting the circles here, like in individual circles, individual parts boxes where the samples are loaded. These samples are not fitting within the right part, right boundaries. So when the computer is reading different cells, see, sometimes the sample pattern is like this, sometimes it is weird pattern like this. or not a circle like this, sometimes small circle, sometimes see abrupt this way. So the system may discard this, calling it it's not as simple as such. Okay, but it may be the required part. So to manage all this, we are using a fixed circle strategy. Okay, so we are putting it like this. I'm only considering analyzing the data coming up from this data. I want only this much data enough. Okay, so I'm calling it as foreground or the background. Okay, I'll only analyze the results or the intensity results coming from this part. Okay, this piece. Okay, I'll not take anything out other than that. So coming up from inside the part, inside the sample, inside the spot is termed as foreground. I want this. All the things coming up from outside is background. Just like I'm doing a colorimetry test, like I subtract the blank reading, okay, from the sample reading. Okay, considering that the blank or the solvent reading will always be same throughout, irrespective of different samples. Likewise, if some intensity is coming up from the outside part, okay, which is abruptly there, drastically varying throughout, I don't consider it at all. I'll only consider this. So even if some value is taken outside, I'll discard it. I'll subtract that value. So I'm calling it as like foreground and the background. Foreground is the one which is coming up from this segment, and the background is the one which is coming up from the outside part. Okay, so this helps me to solve some of the intensity based errors. Okay, now see if this is a slide deck, so the microarray slide deck given here for 5 slides, see, and 5 genes. I'm getting this result, see. Now see if you are getting minus 1.56. See, so for the red intensity, the green intensity, I'm always doing the red foreground minus red background. I'm doing for each sample, each and every sample. I'm sorry. So for each and every sample, I'm doing that. Red foreground minus red background. Likewise for the green foreground minus the green background. Okay, so that everything gets properly done. Then I'm putting the statistical scores and then getting the values. So some values can be plus, some values can be minus here. See? Okay. So then it gives me the heat map. See this way. As I shown you earlier, 10 minutes, 30 minutes, 1 hour, 3 hours, 6 hours, 24 hours. So time course experiments can be done properly here. This is the heat map of the clustergram. This is the resultant clustering, statistical clustering. Because some genes whose expression level is similar, there may be certain connection, okay, certain link. So I can cluster those genes so as to get the functional implications. Okay. So all this this thing is done in statistical clustering algorithms do it. Okay. And these are the programs which are helping me do all this. See, Eisen software or the microarray devices. See, all of them are able to do it properly. Once the image is given, they will be able to give me the fluorescent signal or the data. The disadvantage was the disadvantage of microarray chip. It is relatively too expensive, like less expensive. See, it's less expensive than stage, but still it is expensive. See, $1,000 per slide. This is just an abrupt number. Understand that? I'm just telling you like how much costly it could be. Okay, based on the my different data sources. Quality of data is highly variable. But if you do an experiment for some certain part, now after some time the gene expression levels may change. Okay, so the expression level is depending upon person, depending upon time, depending upon the genetic sources, or depending on what point or what fraction of the part where exactly you are taken the sample from. Several different parts come in there. In each time you may get different data. So what people normally do, they keep on taking the average expression part for this under this condition could be like this. Okay, so too much data is yielded by microarray. Interpretation analysis is highly difficult. See, so see, like after all this, let us discuss the limitations of all the microarrays. First is the dynamic range of gene expressions. See, it's very difficult to simultaneously detect the low and high abundance genes accurately. Even if some gene is not at all expressed, it becomes difficult to analyze. Likewise, if some gene is highly expressed, again we may fail to detect it accurately. Moreover, every gene has got multiple splice variants. Okay, so when the splice variants have got opposite effects. Okay, that is some gene is expressed too much, some gene variant is expressed too less. Okay, we may not be able to quantify the exact implication of all this on a functional part, how the function will be controlled. Translational efficiency is a regulated process. mRNA level does not correlate with the protein level. This is another big part or like a major limitation of a microarray. Even if something is there, we cannot quantify. Even if a bit is there. Proteins are modified post-translationally, glycosylation, phosphorylation, and etc. and all this. Because of which the gene expression part may change. Okay, it has got some part, but we cannot quantify. Now let us discuss some statistical issues coming up with all this microarray part. Okay, first and major one is the planning of microarray. How do you plan? So, design and sample size, all this should be properly done. Like some genes relevant to a given analysis, what kind of genes should I pick up for the probe? That's a big question mark. Okay, then replications of an experiment, as because microarray is able to give me lot much of data every time. So how many experiments or the how many iterations or how many replica plates should I plan? Okay, because it becomes really impossible to achieve absolute independence of replicates. It becomes extremely important. So to plan it properly, because the results may vary. Like per plate results may vary. Depending on different researchers, again the things may vary. Okay. So the experimental error and the human error keep on coming. Okay, then the image analysis part. See, the image analysis again is becomes a major challenge. Addressing, segmenting or quantifying the image data. Okay, because the foreground or the background part or the segmentation error which I've been telling you, that again becomes a major part. Okay, the quality of samples, the log parts, only the dots per square inch, the DPI is very important because I can take the very accurate figure of the complete heat map. Then only I can analyze it further. Okay, normalization is done. I do the statistical normalization, not only within the slide, but also among different slides. Okay, between different slides I do. Then only I can quantify that yes, these genes are present properly, this gene expression is gone rightly. Okay, then biological analysis. The biological analysis part is a major part. After all this is done, now the biological implication part is the most troublesome part here. You need to analyze each and every part properly. Which genes are relatively upregulated, relatively downregulated? The first question. Assigning p-values to the test or confidence results to the results. So that whatever the things are done, I'm confident that yes, they are like this. If some gene is upregulated, I can guarantee that yes, it is certainly upregulated compared to the leftover genes. Or the expression level or average expression level of all the other genes, this gene is highly upregulated. So it has got a functional implication. I should be able to make a complete statement like this with full confidence. Okay. Analysis of time course of the factorial or some other experiments and many more. Okay. But once the data changes, different parts changes, I should be able to exactly quantify all these results. And lastly, the definition and the allocation of various samples. Okay. All these samples. See, I should be able to properly work it out. Now, at the end of this session, let us answer the small MCQ questions so as to check our understanding so far. What is the primary purpose of microarray in molecular biology? Amplify DNA or RNA samples? No. Detect gene mutations? No. Analyze gene expression or protein interactions? The gene expression data is the right answer. Okay. Determine the sequence of our DNA or RNA. So the C becomes the right answer. Which of the following best describes the purpose of microarray? Okay. Microarray is used to generate gene mutations. Microarray provides a method for DNA amplification. Microarray allows for the simultaneous synthesis of thousands of genes or proteins. The C part becomes the right answer. The main principle behind microarray technology is hybridization between target molecules and specific probes. True or false? Okay. Yes, this is indeed true. The major principle behind microarray technology is the hybridization only. Okay, whatever the probe I'm fitting there, until the samples, different targets do not bind properly, I can never get the results. Even if some genes are expressed properly, I cannot quantify until the hybridization is properly there. So only if you remember, we were like exclude not allowing different small individual repeated C's, repeated G's, or like repeated bases, because it may lead to like hairpin loop structures. Okay, because of individual like inverted repeat. Okay, so such kind of binding should not be there. I should be able to exactly get it. Hybridization means the probe and the sample should be able to exactly bind all throughout. Okay, so can it give wrong results? Yes. Okay, if the binding do not take place properly, or if like repeats are there, base of repeats of different bases are there, like the binding or the interaction between the probe and the sample may be abruptly there, not from end to end, but it may be scattered throughout across the chain. So this may give the wrong answer. Okay. So thank you so much for your time and patience. If you have any questions, you can always contact me. Thank you and goodbye.